and get started, I wanted to introduce our speaker for today. So he's come to us from Nottingham in the UK. Just a little bit of background. He received his undergraduate degree in physics in 2002 from the University of Nottingham and did his PhD work at the Sir Peter Mansfield Magnetic Resonance Center at the same university. He focused on arterial spin labeling using the 3T magnet and some beamformer techniques for MEG as well during his PhD work. At the time, there was not an MEG center at Nottingham, so he did some of his work over at Aston University. And then they did get MEG at Nottingham in about 2007. And so he's been helped to set up the MEG lab there and is continuing to also work in MRI using the 7 Tesla magnet there, and is now working really in combining structural information, fMRI, and MEG using beamformer techniques. And with that, I think we'll start with his talk. He's going to talk about his integration of all these techniques. So please welcome Matt.
Thank you very much. Thank you very much for the introduction. Thank you very much for inviting me. This is my first time to Albuquerque. I'm really enjoying myself. So whoever suggested me as a speaker, thank you very much. So as you just heard, I'm from Nottingham, and for those of you who are not familiar with the UK, that's just there. It's quite an old place. It's got quite a lot of history to it. It's had some very famous residents in the past. Not Kevin Costner, he's never been to Nottingham, I don't think, but Robin Hood, he was very unpopular with the local law enforcement. It's also famous for cricket. I'm guessing cricket has not really reached Albuquerque yet, but it's a bit like baseball.
We have some crickets in the back.
But perhaps of more relevance to a sort of neuroscience or neuroimaging community, Nottingham has got quite a history with MRI. So hopefully 1978, so as you can probably tell from the fashion and the facial hair. And for his efforts, Peter, who you saw in that clip, won the Nobel Prize in 2003. The centre in Nottingham now is a really sort of multimodal imaging centre. We've got MEG, we've got EEG, we've got 1.53 and 7T MRI. And so what I really wanted to sort of try and talk to you today a little bit about is majoritively brain networks and the tools that we've now got to probe those networks, so moving from structural imaging to functional imaging. So I'll start with an introduction to networks. I'll then move on to resting state networks in MEG data, so looking at the electrophysiological basis of resting state networks. And then towards the end I'll move on to some task positive as opposed to resting state networks. And if there's time at the end, I'll hopefully tell you, show you some pilot data that we've got on schizophrenia. So over the last sort of 20 years, we've actually become pretty good at doing functional mapping of the human brain. This is a simple example of somatosensory stimulation, where the different colours correspond to somatosensory stimulation of the different digits on the right hand, I think. So this is 7T functional MRI, and you can see that we can really get really nice sort of sub-millimetre functional mapping of the cortex. And this work has been going on for a long time, and it's been very important in terms of mapping the brain. But what I think now is the sort of next challenge for functional neuroimaging is to go beyond simply trying to say that this brain area or that brain area is active or inactive, and to actually start working out what that means. What does the term active, if we say a particular brain area is activated, what does that mean? And how do multiple brain areas actually come together to form to form a network and to undertake a task? So, as I said, I want to focus on connectivity. So, why might you want to measure connectivity? Well, I think we now pretty much know that recruitment of multiple brain regions, multiple functionally specific brain regions, is really key to the way the brain processes information, the way the brain works. We also know, perhaps more importantly than that, that the abnormal recruitment of brain areas can be found in a number of different diseases. So my colleagues in psychiatry tell me that that schizophrenia is a particularly good example of a disease where abnormal functional connectivity underlies many of the observed symptoms. So if we're going to ever go on to understand the brain, we're going to have to understand connectivity. How do these multiple functionally specific brain areas actually communicate? So I'm not a neuroscientist, I'm a physicist, so my interest in this is to try and work out, well, what tools do we really have to probe functional connectivity using neuroimaging? And the obvious one that springs to mind is functional MRI. So over the past, well, since 1995, when Barack Biswell had his first real demonstration of what we now term functional connectivity, where he had a set of resting state fMRI data. And when I say resting state, I mean somebody was basically put in the magnet and asked to do nothing, as much as the brain can ever do nothing. And then what you do is you take a seed voxel somewhere in, say, the right motor cortex, and he looked at what brain areas, what bulb responses from separate brain areas were temporally correlated with that seed in the motor area. And what he found, to his surprise I think, and probably to everybody's surprise, was that he actually pulled out the opposite motor cortex and the supplementary motor area. And that formed a motor or sensory motor network of brain areas. And in many ways what we now term functional connectivity really means correlation between bulb time courses, temporal correlation between bulb responses. Of course, since then, people have not only improved the methods, but also looked and found multiple brain networks. So there are about a dozen different brain networks that we commonly see in functional MRI data, and there's the motor network, there's the visual networks, but also the more sensory networks like the default mode network, the dorsal attention network. And all of these things have slowly become really quite well characterised. But to my mind, at least, the most remarkable thing about these networks is just how robust they are. You can see them in individual subjects, you see them across subjects, you can see them in the resting state, you can see them in the task positive state. And so these things are really, really quite well defined, they're robust, they're very easy to see using functional MRI. But anybody who's ever done functional MRI has probably seen either this diagram or a diagram very similar to it. The problem with fMRI is that you're measuring an indirect response to brain activity. So what happens? Well, in a sort of classic neuroimaging experiment, we might show somebody some sort of stimulus, so a visual stimulus. That induces a neural response. There's also a chemical response, so neurotransmitters are doing their thing. Those two things together have an increased energy demand, and so there's a vascular response. There's an increase in cerebral blood flow, increase in cerebral blood volume, and an increase in the uptake of oxygen. And the increase in CBF, CBV, and CMRO2 all combine in some clever and convoluted way to give you the bulb fMRI time course. And that's right over here, and what we're probably interested in is what's going on at this level. So the bulb response, fantastic for spatial mapping and wonderful spatial resolution, but it's an indirect measure of brain activity. It can't measure the electrical signals that are inside the brain, and it's got relatively poor sort of 5 to 8 second temporal resolution. So going back to what tools do we have to really start probing functional connectivity, perhaps one obvious way to go is from fMRI to MEG, because it's got certain advantages. It's a more direct measure of neuronal activity. And by that I mean that we bypass the hemodynamic response and we measure the magnetic fields that are generated directly by electrical activity in the brain. And in this context, electrical activity means synchronous firing of large clusters of cells. So that's a good thing. We've bypassed the hemodynamic response. It's got great temporal resolution, sort of millisecond time scale. So that's good. So because we've bypassed the hemodynamics, we don't have to wait for a hemodynamic response to brain activity. So great temporal resolution. The spatial resolution is limited because it's an ill-posed inverse problem, and we'll cover that later on. But the spatial resolution is probably better than EEG. And this, to my mind, makes MEG the most attractive non-invasive electrophysiological measurement for probing functional connectivity. It's also got whole brain coverage. So on the face of it, things look pretty good. But of course, as with everything, for all the advantages, there's a set of disadvantages. And we can go through those. Magnetic field spread means that there's no one-to-one relationship between the sensors and the sources. So one single source will affect multiple sensors. And depending on which MEG system you have, probably the most affected sensor is not necessarily the closest sensor to the source. That makes sensor-space measurements of connectivity pretty difficult to interpret. But then you add to that, so you can project your sensor space data into source space using some sort of inverse solution or source localization, or call it whatever you will. And people sort of might think, well, if we project it from sensor space to source space, we've actually sorted out a lot of those limitations. But that's not true, because in source space you get leakage from one voxel to another voxel. So unlike fMRI, where voxel measurements are pretty much independent, in MEG, neighbouring voxels are very much, the temporal signals from neighbouring voxels are very much correlated. And that source leakage is not symmetrical about the source, or it's a sort of very asymmetric, difficult problem to deal with. MEG's also susceptible to interference. It's got a pretty low signal-to-noise ratio. So let me try and expand a little bit on what I mean by magnetic field spread, first of all. So we've got two simulated sources here, one in the right motor cortex, one in the left motor cortex. We then put some MEG sensors around. These are the sensors that we have in Nottingham. This is the CTF system. And then if we switch on those two sources, we'll get some magnetic fields over here and some magnetic fields over here. Now in functional connectivity, what we're hoping for is that those two sources will in some way communicate with each other. They'll be co-modulating. And so we have to add them together. And what we end up with, hopefully you can just about see this, is something in sensor space that's actually pretty difficult to interpret already. And that's the perfect situation with only two sources. So rather than playing with simulations, let's try and do this for real. So this was the first experiment that we started doing on functional connectivity, and we did these in around 2009, maybe 2010. And all we did was exactly the same as Bharat Biswal's experiment. We recorded 5 minutes of resting state MEG data. And again, when I say resting state, people were lying in the MEG system, and they were told not to do anything, and as far as possible not to think anything. So we recorded our data, we frequency filtered our data, and what I'm going to show you is MEG data filtered into the beta band. We then did something called Hilbert envelope computation. And to cut a very long story short, if this is a beta band oscillation, this black line here, then the envelope, the Hilbert envelope of that beta oscillation is this red line here. What we then did is something called seed-based correlation. So very similar to the original Biswal study, but this time in MEG. We took a single sensor, which was over the right motor cortex, somewhere over here, and then we computed the Hilbert envelope for that sensor, and then we looked at temporal correlation between that seed sensor and the Hilbert envelopes of all of the other sensors surrounding the head. And what we got is spectacularly unimpressive. Here's the seed sensor, which, as I said, is roughly over the right motor area. And what you see is that by far and away the most correlated sensors are those that are immediately surrounding it. And this is this idea that one source affects multiple sensors. But the good news was, if you actually look over here, you can see this sort of dark blackish area, which has got a correlation coefficient of about 0.5 or 0.6. And that's implying that at least there is a degree of cross-hemisphere temporal correlation between these envelope signals. So that gave us some encouragement that what people have been seeing in fMRI for years could also be seen in MEG by considering the sort of envelope correlation. And we weren't the first people to do this. One of the first papers that I know of was actually by Jess Dunn's group in NIH. But these pictures are particularly hard to interpret, so what we did was move from sensor space into source space. And the way we did that was using a beamformer approach to solving the inverse problem. So I guess some people may be familiar with beamformers, some people not. So those of you who were here yesterday, sorry, the next 4 slides are going to be exactly the same as I showed you yesterday. A beamformer is a means to project from sensor space to source space. And the way you do that is you first of all pick a location inside the brain, any location you feel like, and you say, right, the electrical signal that's going on at that particular brain location is going to be given by a weighted sum of the magnetic fields, a weighted sum of the MEG sensor measurements. And this is a general formulation, and if you want you can write down any inverse solution in this way. So our electrical signal is our weights times our data. The clever bit is how do you define the weights? And that's where the inverse solutions differ. Beamforming uses this thing called power minimization or variance minimization. So what we do is we base the weights on a minimization of the output signal variance. So we minimize the variance of this thing that I've called Q. Now, of course, if you just derive the weights based on minimizing the variance, you'll get 0 weights and you'll get 0 variance. And so that would be silly. So you have to put in a linear constraint, and the linear constraint is that power originating from your chosen location, that one location in the brain that you have chosen, must remain in the output signal. So you can actually formulate that mathematically, and I won't go into the details, but you end up with this, and the nice thing is that that particular equation, that mathematical form, you can solve using Lagrange multipliers. And you can solve it and you can just get an equation for the weights. And the weights end up based on 2 things. They end up based on a lead field model. So that's a model of what magnetic field you might measure if there were a source at your chosen location. And the C in that equation is the data covariance matrix. So that's a measurement of the covariance between 2 signals measured at 2 separate sensors. So we can derive the weights, and we can derive the weights for any location in the brain. And what we can do is we can define a set of voxels based on some grid, and we get weights for position R1, R2, R3, and so on. So for any one location, we can get an output time course. So we can get a time course of electrical activity for a particular location in the brain. And in this case, what I'm showing is a time course of electrical activity from the visual cortex when somebody's watching a flashing checkerboard. And it's a bit hard to see, but there's an oscillation in here that's going at sort of twice the flashing checkerboard frequency. You can then also do this sequentially on a number of voxels, and if you come up with some imaging statistic, like you might say, well, I'm interested in the difference between this time and this time, then you can make an image and you can work out where in the brain something was happening. And that's basically beamforming. So going back to our connectivity, this time we're going to do it slightly differently. Rather than looking in sensor space, we're going to look in source space. So the first thing you do is you generate your covariance matrix, you generate your lead fields, you take all of the data that you've got for any particular subject, and you project that through the beamformer spatial filters and into the brain. Then for your brain space, your voxel level signal, you compute a Hilbert envelope again in exactly the same way as we did before. So we've got, remember that the data are frequency filtered, so we've got beta band data that we've beamformed, we've computed the Hilbert envelope, and again we can do seed-based correlation, but this time rather than picking a seed sensor, we can pick a seed voxel in exactly the same way as you would do with seed-based correlation in fMRI. And so we pick a seed, and then we say where in the brain is functionally connected to that seed. Now, if you do that in the motor network, what you find is that in this case the seed is on the right-hand side of the image, where that big, horrible-looking yellow blob is. And you find the most connected area outside that general region is actually in the left motor cortex. Okay, so this is a good thing. It's showing that our MEG data, our electrophysiological data, can show us the motor network in exactly the same way as functional MRI can show us the motor network. It doesn't look as pretty as functional MRI, and that's because of this large seed blur. And the large seed blur is coming courtesy of the ill-posed inverse problem. And if we just compare the sensor space measurement to the source space measurement, at least I think this is rather easier to interpret than that. Here we're actually getting some spatial information. It's a smooth map, but we're getting some spatial information about, yeah?
So do you get any SMA localization or cerebellum localization?
We tend not to get the SMA. Or I should say, we get the SMA in about maybe half of our subjects routinely. And there are various possible reasons for that. The reason that's usually given is that you get opposing dipoles, which actually act to cancel out some of the MEG signals, some of the magnetic field that are being generated by the SMA. But it's an interesting question, and I've spoken to several people about it, and some people say, yeah, we see it in half our people, 3/4 of the people, 1/4 of the people. It's something that somebody needs to look into, I think.
You have some midline sort of signal on the left map on the top there, right? What does that?
In this?
Yeah.
That's probably due purely to volume conduction, I would guess. So in general, doing it this way is a much better idea than doing it the other way. The other real advantage that you get from doing source localization, and I don't want to go into any detail now, but if anybody's interested they can ask me later, is by doing source localization, it's a really effective way of eliminating interference. So remember that the beamformer is trying to minimize the variance of anything that doesn't look like it's coming from your locations of interest inside the brain. And so things like the heartbeat artifacts, respiratory artifacts, artifacts from your computer equipment that's sat outside in the lab, all of those things are actually quite effectively suppressed by the beamformer. And so, so there are, beyond this, which is, you know, the main advantage of going into source space, the fact that you can actually see in the brain where you're looking at, you also get quite a nice artifact rejection. But I won't go into that too much. So here we have this same stuff now, this time on a subject group. This is, I think this was 20 subjects when this is quite old data now. When we first started, at the top we've got the fMRI network, and here we've got a seed in the left side of the image, and that pulls out something in the motor area in the right hemisphere, and here if you put the seed in the right hemisphere, sure enough we can pull out something in the left. So what this is showing us is that, at least in the case of the motor network, we can get sort of qualitatively similar networks. We can extend that a little bit, so you might want to go for, this is variously known as the frontoparietal network or the dorsal attention network. It's usually split into left and right, and here it is in fMRI. So here the red overlay shows the left frontoparietal network, and the yellow overlay shows the right frontoparietal network. And sure enough, if we play the same game, seed-based correlation, we put a seed here, we pull out this frontal area here, we put a seed the other side, we pull out this frontal area here. Again, you got horrible seed blur. This whole region of brain is coming up, and that's spurious connectivity caused by just the signal leakage in MEG. But there is a general structure in there that's starting to suggest that you can indeed see some of these networks. And you can do that with a number of networks.
How large is the seed?
Pardon?
How large is the seed?
The seed is a single point.
A single point?
Yeah. But of course, you'll get a certain, with the beamformer, you'll get a certain beam width, and you can actually characterize the point spread function of the beamformer. So it's not like fMRI where you say the seed is 3 mil cubed. It's the seed is actually defined by the signal to noise ratio of the data. So it would be nice to get away from this sort of horrible seed blur and to try and do something to try and help out the algorithm. And so what we did was to do what I seem to have been doing for a long time, which was to find out what the fMRI guys did 5 years ago and just do that. So with that in mind, we did some independent component analysis. So again, we've got step one, which is beamform from sensor space to source space, and again, most of the stuff I'm going to show you is beta band. Then we do our Hilbert envelope computation again, and then we do, this time, temporal ICA. And what we're looking for is envelope is temporally independent time signatures, for want of a better word, that exist within the data. So we're looking for groups of voxels in source space that have the same independent temporal signal within their within the measured Hilbert envelope. And of course, once you've found those temporal signals, you can work out from where in the brain those temporal signals originate. And that gives you clusters of voxels. And then that gives you a set of networks, and you need to work out which networks you want to keep and which networks you want to throw away. And so what we did was we compared the networks that we got in MEG to the networks that we pulled out of functional MRI data to see which ones looked spatially similar. And this is what we get. So in all cases, the fMRI network is at the top, and the closest MEG equivalent is underneath it. And so you can see with the left and the right frontoparietal networks, we get something that looks like the motor network, something that looks a little bit like DMN. Should point out that DMN was in the alpha band, and all of the other networks that I'm going to show you were in the beta band. And we can sort of carry on. There was 8 reasonably good matches. There was this sort of frontal thing that was going on. There was a medial parietal thing. There was a visual network, and there was something going on that looked like the cerebellum. So these are not perfect matches by any means, but they do imply that similar networks that we see in fMRI data can also be observed using MEG data if we look at the Hilbert envelope. But there's a slight problem with looking at the resting state, and that is you're never quite sure what the brain's doing. And you can tell somebody to lie in a scanner and do nothing and think nothing, but I mean, if I tell you to think nothing, what do? You immediately start thinking of something. So much as there's a whole literature on the resting state, it is actually pretty hard to really nail down what some of these fluctuations in the Hilbert envelope actually correspond to, and how many of these, yeah?
Just a quick question now. When you were talking about the Hilbert envelope, did you actually look at the specific bands, like alpha versus beta versus gamma?
Yeah. So you do. We tend to split the data. So most of the stuff I showed you was beta, apart from the DMN, which was alpha. So we tend to split it into delta, 1 to 4 hertz, theta, 4 to 8, alpha, 8 to 13, beta, 13 to 30, and then a sort of low gamma, 30 to 50. We've also tried going higher, but the signal to noise, when you get really high, is actually quite low. So, yeah, but again, that gives you another problem, that you can derive 15 or 20 independent components for the beta band, but then you've got another 15 or 20 for the alpha band, another 15 or 20 for the theta band, and so on and so forth. So all of a sudden you've got a hell of a lot of components, and you've got to find some way of working out what. Yeah?
Just out of curiosity, do you see any differential in the connectivity with MEG dependent on the band that you're looking at? There's been a lot of talk about alpha and default mode and gamma default mode and motor differences, or do they do all of the components share similar relationships regardless of the band?
No, you get different components out depending on the band. So the ones that best match the functional MRI networks actually come out in the beta band. But some of those are also reflected in the alpha band, some are reflected in the theta band, but you get different components. As a rule, you get different components in the different bands. And so it's a sort of open question as to how you best combine those bands. And we've had a bit of a go at it using a technique that we're sort of calling meta-ICA. But, well, we can talk about that afterwards if you want. Okay, so with resting state networks, it's difficult to know what's going on. And so what we actually tried to do was to introduce some tasks. And the fMRI literature has shown that we get very robust networks, these same networks out, whether or not there's a task going on or not, pretty much. They do change a little bit, but by and large their spatial structure is very similar. And so we wanted to see if we could get similar networks in a whole new task-positive data set. If we can, then how do the neural oscillatory signals within those networks, how do the alpha band or the beta band or the gamma band signals within those networks change with respect to those tasks? And can we actually use that in some way to look at the sequential, temporally sequence the activity across the different networks? Because that sort of measurement, that sort of what you might term effective connectivity, working out which network fires first, is pretty is pretty confounded in fMRI, because the hemodynamic response changes depending on where you are in the brain. So if you look at two different brain areas, the hemodynamic response can be shifted by half a second. So with that in mind, we used our CTF system again, and we had three tasks: a Sternberg, an Enbach, and a relevance modulation task. I'm guessing quite a lot of people are going to have heard of Sternberg and Enbach, but just to go through what those things are. In the Sternberg, what people were looking at were two visual stimuli, and these were sort of arbitrary shapes. They didn't really look like anything. They were designed not to look particularly like anything, but they were arbitrary shapes that people saw. So you saw two of those that were presented in very quick succession, about a second between them. Then there was a 7-second, I think, maintenance phase, and then you saw a third shape. And what you had to do was press a button if that third shape matched either of the first two. So it's basic working memory task. you try and remember the shapes. It's really straightforward. In the second task, this was an NBAC task, so here people saw a sequence of letters, one letter every 2 seconds, and you had to press a button if the current letter matched the letter that was presented n letters previously, which is a long way of saying if it's a 1 back and you get an A and a B and a B, then because 1 letter back was also a B, you press the button. If it's a 2 back, we get a D, a G and a D, so this becomes a target. The third task was what we called the relevance modulation task, which was a bit more complicated. But basically what we had was an example visual stimulus, which was either a picture of a butterfly or a ladybird. Okay? And then we had a series of relevant stimuli and a series of irrelevant stimuli. So if the example was a picture of a butterfly, then all of the relevant stimuli would be pictures of butterflies. And you had to press a button if any one of those relevant stimuli, any one of those butterflies, matched the example. If the example stimulus was a ladybird, then all of the relevant stimuli would be ladybirds, and you had to press a button if the current ladybird matched that was presented at the example. And the irrelevant stimuli were sort of distractors. So what we were looking for was differences in brain network oscillations for relevant and irrelevant stimuli. So we've got Sternberg, NBAC, and relevance modulation. What we did was we collapsed all of those tasks together, which gave us about a total of about 10 hours worth of task-positive MEG recorded across 28 subjects and all 3 tasks. And then we used the same or very similar independent component analysis technique to pull out the networks. And these were the networks that we got. Again, you can see sort of motor areas, the visual areas, and you can see the frontoparietal thing. This time it's joined, it's not separated left and right, it's actually joined. You also get bilateral insular somewhere there. So we got a handful of networks that sort of looked physiologically plausible. And what you can then do is, because it's rather than the sort of the resting state stuff where you get the networks and that's it, you can actually go and probe what those networks are doing during those 3 tasks. So if we look at the Sternberg as an example, you can see that the visual network is responding to those first 2 stimuli that are presented. So if you remember, there's the 2 stimuli, then the maintenance phase, and then the 3rd stimulus. So it responds to the first 2 stimuli, then it does nothing for a little while, and then it responds to the 3rd stimulus. And the lateral visual areas, well, they do something very similar. But then if we go into areas of the brain that you might consider as part of the more attentional networks, like the frontoparietal network or the default mode or the dorsal attention network, frontoparietal network, what you see there is, rather than single isolated responses to the visual stimulus, you actually get a prolonged beta band desynchronization throughout the maintenance phase, and that's true of these 2 more attentional networks. So that's the Sternberg task, and obviously you can look at any number of different networks. We see the left motor area coming out, and you can see this is sort of when people responded and they pressed the button. You've got the characteristic beta band desynchronization and the beta bands. If we then look at the NBAC data, again, the first thing that you can note here is these time frequency spectrograms are a 30-second block of either 0 back, 1 back, 2 back, or 3 back. And obviously what you could do is you could zoom in to really exploit the temporal resolution of MEG to look at the responses to each individual button press. This is rather sort of looking at it collapsed across the 0 back, 1 back, 2 back, or 3 back phases. And what you can see is, again, you can see in the visual cortex the responses to each individual letter. But if you go into the attentional networks again, here you can see, well, there's not much going on at 0 back or 1 back, but 2 back you start to see, again, beta desynchronization, and 3 back, again, beta desynchronization. And in the frontoparietal network, you can see a beta desynchronization and a theta band synchronization. So there's quite a lot going on in these networks. This is the relevance modulation task, and again we've got all the networks. And this side we've got the response for the relevant, and this side we've got the response for the irrelevant. And you can immediately see that even in the visual areas, that there are actually differences between a relevant and an irrelevant stimulus. And then again, when you get to the attentional network, you can see quite big differences in the time frequency evolution of the oscillations. So, what this is giving us is a way to collapse, say, 4,000 or 5,000 voxels down into a handful of networks, all of which exhibit some similar pattern in terms of their Hilbert envelopes, and then a means to actually go back to the full-time resolution of the data and say what was going on in those networks given some task. Okay, so in the last 5 minutes, I'm going to just finish by talking about some pilot work that we're doing. I should say this is we only started this project in July, and so far we've managed to scan 24 people, 12 patients with schizophrenia, and 12 controls. And the controls were matched for age, sex, and socio-economic status, but not IQ. All of our patients were on some form of medication. So the results that I'm about to show do come with a lot of caveats, basically. But our aim was to use the Sternberg and the relevance modulation test to look at network oscillations in schizophrenia. And we had a number of hypotheses that I don't take any credit for. My psychiatry colleagues, in particular Peter Liddell in Nottingham, he came up with several hypotheses. I've just listed 2 here. He said that the task-induced modulation of oscillations in the attentional networks will be different in patients relative to controls. And perhaps more specifically than that, in the relevance modulation task, he thought that the difference between the responses to relevant and irrelevant stimuli would actually, those differences between the 2 different stimulus types would be much less pronounced in the patients with respect to the controls. So in other words, just going back to this, these differences that we see here, he was suggesting would go away when we started looking at the patients. So these were the initial results from a group of 12 controls and 12 patients. And the first thing that you know is, apart from the fact that my PhD student can make much prettier pictures than I can, because these are much smoother and interpolated, what you immediately see is that actually if we take the insular network, for instance, this sort of theta synchronization and then a later beta synchronization in the relevant that is sort of not matched in the irrelevant case, in this group of controls, is rather similar to what we saw in the initial group. So these are different people, but using similar methods. So we've got quite a good agreement between what we saw in the first set and what we saw in the second set. But then if we go and look in the patients, what do we find? Well, first of all, even in the visual areas, you can sort of see that there's a difference. You get this prolonged beta desynchronization in the patients that doesn't appear to be there in the controls. And just as my psychiatry colleagues predicted, the difference in patients between relevant and irrelevant is much less pronounced than the difference in controls between relevant and irrelevant. So there are some interesting features, even though this is a very, very small data set. Oops. Yeah. This is patients and controls of the Sternberg, and again, it's perhaps less interesting, but what you can see is, in general, there's more prolonged beta desynchronization in the patients than there is in the controls, and again, you can see in the visual areas, in the same way as we saw an extended beta band desynchronization for the relevance modulation task, you can also see extended desynchronization in the Sternberg task. And I'll finish with this observation, and again, these are very small patient numbers, so you can take this with a pinch of salt, but in general, what we've plotted here is theta band amplitude during the maintenance phase of this Sternberg task versus, in this case, accuracy in the task, and in this case, reaction time. And what you can see is that those patients who are better able to suppress their theta band response, in general, were more accurate and had faster reaction times. So, to try and sum up, I think fMRI probably remains the technique of choice if what you want to do is spatial mapping of networks. But that said, similar networks to those that you can see in fMRI are available in MEG data if you look at the Hilbert envelope. Now, the Hilbert envelope is probably not the only way of doing it, it's probably not even the most interesting way of doing it, but it does work. If you can measure functional connectivity in sensor space, but the results are actually pretty difficult to interpret, and moving to source space gives you a massive advantage in terms of not only the interpretability of your data, but also, perhaps more importantly almost, the reliability of your data, because source space projection will significantly improve your signal to noise ratio. We can do this in the resting state, but it's arguably more interesting to do it in a task. And we can look at network oscillations, and we can see what they're doing in response to these various different working memory tasks. And significant changes in network oscillations can be measured in response to all of the tasks that I've presented. And finally, I think there's an awful long way to go with the schizophrenia project, but the initial findings appear to show that this could be quite an interesting area for future study, and I think that's the only conclusion I feel confident in drawing so far. Once again, thanks for the invite. It's been great coming over and meeting all you guys. Thanks to all of these people, and thanks to you for listening. Yeah, so in general, so the part that perhaps I missed out, perhaps I shouldn't have missed out, was the, unlike the case with the resting state networks where the ones I presented were all in an individual band, these particular networks were actually defined by collapsing across all of the frequency bands using this technique that I sort of mentioned called meta-ICA. And that's why they look a lot more blurred, because they're collapsed across many more components. But in general, the finding is that the most similar networks to those that we see in fMRI are usually in the beta band, with the odd exception of the default mode network, which variously appears in either delta, theta, alpha, or all three. So in general, most of the others are pretty robust, pretty easy to get. The default mode network remains slightly elusive. My collaborators in Oxford say that the best way to do it is by trying to collapse across frequency bands. But yeah, the beta band is probably the most useful.
I've never worked with MUT, so probably question is very naive. So you mentioned that spatial resolution is, for me, it's hard to describe, or what is the spatial resolution?
So spatial resolution in MEG is exactly the same as it is in fMRI. So do you work with fMRI?
Right.
So in fMRI you can say, right, I've got a voxel and it's 3 millimeters cubed. Yeah. In MEG you can also define the size of not a voxel but a resolution element. Okay. So you can say my signal that I'm interested in, that I want to be coming from this particular location, is actually coming from this volume of tissue. Okay. The difficulty is that volume of tissue is not a nice cube, it's not even a sphere, it's some sort of horrible asymmetric looking thing that will entirely depend, in the case of beamforming, on the data and where in the brain you're looking. So yeah, spatial resolution in MEG, it sort of means the same thing. It's, you know, how close can I bring, so a physicist's interpretation of spatial resolution is how close can I bring two sources before I can't separate them anymore. And that's what it effectively means. And but it's actually quite an elusive concept. It's actually quite tricky to measure. There are ways to measure it, but it's quite difficult.
Wouldn't you agree that the MEG's resolution looks to be a little lower in terms of smoother? Or
than fMRI?
Yeah.
Oh, absolutely.
Okay.
Yeah. If you go back to the actual comparisons and take the case of the motor network, for instance, I mean, you look at this and you look at this, and this has got much better spatial resolution than that. And so if spatial resolution is your thing, if that's what you want to measure, if you expect in your group of patients with whatever syndrome that it's going to be a spatial difference in your networks that you want to look for, then use fMRI, don't use MEG, because the ill-posed inverse problem means that it's got much lower spatial resolution. But if it's temporal information that you're after, or you want to, for some reason, bypass the hemodynamics and get straight to neural oscillatory signals, beta band, gamma band, whatever, then MEG can provide similar information, just at lower spatial resolution.
Your comparison between the control and schizophrenic, the Sternberg test, is there anything to be read into that apparently larger response in the schizophrenic?
You mean, which responses are you talking about? The beta band desynchronization? It's a good question. I hope so. I don't.
Consistent?
It is consistent, and it is, at least according to all of the statistical tests that we've tried in 12 people, it is statistically significant. There is a statistically significant larger response in the patients than the controls. But as I said, this is a small group of patients. There was, in general, as with all of these studies, there's more variability in patients than there is in controls. They move around more. And so I personally think, so I put these data in because I knew there were people who were interested in schizophrenia here. I personally think, as I said, the only conclusion that I'm drawing so far is that I think this is interesting, and I think probably these things are real, or at least I hope they're real. But time will tell. Yeah.
I could just raise the question, can you from animal studies where you can actually get into the neural networks as well as doing something on the outside with this, so much energy to say, what is it that's driving beta? What is it that's driving gamma?
Yeah. Well, I was saying this morning actually what I think would be really nice. So we don't do animal experiments in our centre, but I think what would be actually quite nice is for somebody to go and have a look at these envelope correlations using invasively recorded electrophysiological data, and say, first of all, do these same envelope correlations in the networks exist in the brain of a rat, where we know we can do fMRI and get analogous brain networks with the bold response. So do these same beta band oscillations and beta band envelopes still correlate in that case? And then what's really underlying that? So there's this theory to suggest that it's when you get an increase in beta band envelope, underlying that might be some sort of synchrony between the underlying beta band signals. That's actually pretty difficult to probe in MEG because the signal to noise is fairly low. Now, if you could do that in a rat using electrophysiology, you would have vastly more signal to noise. So it would be actually great if somebody would do those experiments, I think. Yeah. Sorry. Yes. Go.
Let's go back to spatial resolution.
Uh-huh.
I heard you say with MEG how far apart do two sources need to be before you can separate them.
Yeah.
And if you think about the way fMRI defines it, usually it's voxel size.
Yeah.
And so if you look traditionally at originally and MEG, defined it more in terms of how far apart two sources have to be to differentiate between them. But that's never been really applied to fMRI. So how come they're able to get around that definition?
Well, I think everybody in fMRI, I'm no expert in this. The person who should be answering this question is Sue Francis, who's one of my colleagues in Nottingham. In general, I think people in fMRI assume that if you've got two independent voxels, that means two independent signals. And of course, there's no spatial bias, unless you're using parallel imaging, in which case there's a bit of spatial bias. But if you're using a bog standard echoplanar imaging sequence, there should be no leakage between voxels in the same way that there's leakage between voxels in MEG. But at the same time, there is an inherent point spread function in fMRI that's actually caused by the tissue itself, motion and whatever else. And so, I mean, as I said, as a physicist, the idea of spatial resolution goes back to something like the Rayleigh criteria and how close can two things be before they look like one thing. And that's always been my view on what spatial resolution means. So were I to measure spatial resolution in fMRI, which I've never done, if I was going to, that's how I'd do it. Yeah.
It seems like you'd have to do it experimentally to define, like do you actually have a visual space, a source in one location, then a source in another, and could you, with fMRI, differentiate between them if they're close? But how far apart do they have to be?
That was the first slide, that was the homolog regions for sensory mapping that people have already shown. I mean, other studies in 7T showing, individual digits and face areas, so you're getting this really exquisite temporal or not spatial resolution of the medial cortex corresponding to the no homolog areas for the face, the hands, and the foot. So
it's very high,
yeah.
I would argue it depends on what you're...
And also the visual system.
Yeah, but it depends on what you're measuring. I think people doing fMRI, I can't speak for everyone, but I know a lot of people doing fMRI use that same definition, full width half max kind of separation criteria, and if you're looking at bold fMRI, then that's a different thing than if you're looking at anatomical MRI.
Well, and I'm specifically referring to bold.
Yeah, and so, even the Upburn study, they looked a lot at smoothness, right? So they're estimating smoothness on different scanners and quantifying that as a way to try to standardize across sites. And so I think there's a lot of this already out there in the fMRI community that sort of looked at that as one of the measures. So.
Well, but I haven't seen anybody really do a systematic study.
But the ultimate fact is the resolution of fMRI, as you said, depends on can you pull apart these different areas, and certainly it ultimately probably depends on how closely the brain decides to regulate blood flow to different areas, and all the evidence is that that's incredibly spatially specific. We can do different digits, which I showed. We can do different areas of visual cortex, and there's even these studies now looking at orientation columns in in the visual field, isn't there? And showing so the spatial resolution of fMRI is really very high, particularly as you go to higher and higher field.
But we don't really know what the spatial resolution of MEG is. If you look at some of these different, like beamformers, for example.
Well, the spatial resolution of MEG in beamforming will depend actually on the data. It depends on the signal to noise of the data. And I think Joachim Gross did some stuff where he looked at the full width half max of a beamformer point spread function, and it turns out that it's basically a function of signal to noise ratio of your data. If you've got really high signal to noise ratio and only one source in the brain, it's really very high. In studies like this, where you've got sort of it's resting state data, there's no one source that's synchronizing and desynchronizing, and the spatial resolution will be inherently lower, and that's why you get some blurred maps of the network.